Again, welcome everyone to today's webinar, and thank you so much for taking the time to join. The webinar today is presented by us at Techtum Lab, together with our partners, BioSkryb Genomics and S2 Genomics. And the topic, as you've seen in the invite, is from tissue sample to comprehensive genome and transcriptome data. So here's a short overview of the agenda. So I will start with just the 2 slides, a quick introduction about Techtum Labs for those of you who are not familiar with us. And then I will hand over to Nabia from S2 Genomics to talk about automating tissue dissociation for single-cell analysis. And then after that, John from BioSkryb Genomics will talk about a revolution in single-cell genomics featuring ResolveOME and ResolveOME. And in the end, we'll have a Q&A session. So if you have any questions during the presentations, please write them in the chat, and we'll make sure to address any questions in the end. So a quick presentation about Techtum for those of you who do not know us. I'm just quickly gonna let in people who are waiting in the lobby. Sorry about that. So. Back to Techtum. Techtum is a full-service partner to molecular biology labs in the Nordics. So we offer complete solutions, including service and support, to our customers that are both in academia and in the industry. Techtum has actually been in the market for over 45 years, and soon 50, because next year we celebrate 50 years, which is of course something we are proud of. And Techtum is available in the Nordics, in all the Nordic countries, but we have our main market in Sweden. And why we want to highlight that we are a full service partner is because we both have our experienced product specialists that can support customers through the purchasing journey, and then after that with technical support. Then we also have service technicians who can provide aftermarket service and repairs of instruments that you purchase from us. So just a high-level image of what Techtum offers. So we like to focus on not just products, but actually on solutions and workflow that enable our customers to improve their lab experience and overcome obstacles. So we focus on different kinds of solutions for molecular biology labs, all the way from sample collection, extraction, and PCR solutions into what we're going to talk about today, regarding single-cell and NGS analysis. So with that, I'll take it back to the topic of the webinar, and we're going to talk about how we can go from a tissue sample to comprehensive genome and transcriptome data from an individual cell. And with that, I will hand over to Nabia from S2 Genomics to talk more about their solutions and how they can improve your lab workflows. So with that, I'll leave the screen to you, Nabia.
Hi everyone, I'm just pulling up my window here. Can everyone see my screen?
Yes, I can see it.
Perfect. Well, hello everyone, good afternoon, and for those of us joining here in the States, good morning. I'm Nabiha. I'm a product manager here at S2 Genomics, and I'm excited to present how the Singulator platform can simplify sample preparation and deliver reproducible results. We all know that biology, so I kind of want to just go over a basic overview. So we all know that biology is immensely complex and that each organ is made up of many different sample types or cell types. One recent advancement in genomics is the adoption of single-cell genomics assays, which gives scientists ability to study transcriptome and epigenome at the single-cell level in millions of cells simultaneously, allowing us to measure cell types and diversity at an unprecedented scale and resolution. So single-cell genomics has transformed our understanding of biological systems. It offers not only high-resolution insights into cellular diversity, but helps us understand cellular states and functions, enables the discovery of new cell types, so on and so forth. Single-cell approaches are invaluable for studying things like disease mechanisms, identifying therapeutic targets, understanding epigenomic regulations, and supporting integrative multi-omic research. However, high-quality single-cell suspensions are not always attainable. For complex samples, nuclei isolation has distinct advantages. Nuclear preparation enables better discovery of embedded cell types and is ideal for tissues with low cell viability. It also reduces bias towards immune cells and avoids enzyme-induced stress and transcriptome alterations, and the single cell method may introduce. So whether you're looking for obtaining cells or nuclei, choosing a dissociation method is crucial, and there are many factors to consider. Some of the things to consider are the mechanical method, because some methods can cause severe damage to the cells or nuclei. Whether you will be conducting a chemical lysis for nuclei isolations or an enzymatic dissociation for cells. You will also need to determine the debris removal method, since debris can cause run failures or high levels of background reads in your sequencing data. It's also best to know what you're looking for, because if you're looking for cell surface epitopes, for example, then you will want to conduct cell isolations in a way that won't cleave them off. With that, there are common methods in dissociating your sample, and one common dissociation method is manual sample prep, and it is also one of the main bottlenecks in single-cell analysis. In this process, preparation of the tissue, buffers, the dissociation, filtration, and even determining the debris removal method is all done by hand and requires researchers to piece together their own protocols. This makes manual sample prep time-consuming, introduces technical variability between one researcher to the other, and can lead to inconsistent data. Similar to manual sample prep, a semi-automated sample prep process still requires many hands-on steps and is time-consuming. Each step introduces an opportunity for human error, and these debris removal steps remain very cumbersome and can lead to sample loss. These methods are expensive because each step requires supplies from different vendors. Even within a single vendor, complete protocols are usually not available, which means that the researcher still needs to piece together their own protocols. So at S2 Genomics, we recognize the benefit of a complete, streamlined, automated solution. With the Singulator platform, you can now automate your sample prep. The platform isolates viable cells or intact nuclei, delivering a streamlined, automated sample prep method with minimal manual intervention. Using our Singulator platform, it's as easy as placing your sample in a cartridge and initiating a run. Our custom reagents, cartridges, and protocols not only save time, but provide high-quality cells and nuclei suitable for many downstream applications. And how it works is quite simple. Users collect their fresh, frozen, OCT embedded, or FFPE tissues. They place them in a cartridge, select the protocol, and press run. And within an hour, you have your intact cells or your nuclei, and not just one sample, but many. And we'll go over that in a second. But what makes a high-quality single-cell suspension? High-quality single-cell suspensions have minimal cell clumping, high cell viability, and low debris levels, which improves the accuracy of cell analysis and profiling and sequencing results. Achieving high-quality single-cell suspensions is crucial for downstream analysis. So let's take a look at kidney cells isolated on the Singulator platform. Like many organs, the kidney is made up of different types of cells, which we observe here stained in trypan blue. We also see in this isolation favorably high viability, indicative of the lack of trypan blue inside the cells, minimal debris, minimal aggregates, which can be seen as the cells are well singulated, and optimal concentrations, enabling the sample to be taken into any downstream application. And so then what constitutes as a high-quality single nuclei suspension? A high-quality nuclei suspension should include intact, debris-free nuclei that retain a smooth, round shape, as well as intact heterochromatin structure supporting reliable gene profiling and chromatin accessibility. So now let's take a look at liver nuclei isolated on the Singulator platform and stained with trypan blue. We see in this isolation that the nuclei have that round and smooth intact nuclear morphology, minimal debris, minimal aggregates, which again can be seen as the nuclei are well singulated, and optimal concentrations, again enabling this sample to be taken into almost any downstream assay. The Singulator achieves high yields and viabilities from cell isolations across various tissue types. Depending on the expected cell density of the tissue, you can observe cells anywhere from around 5,000 cells per milligram of tissue to hundreds of thousands of cells per milligram of tissue with viabilities of 80% or higher. Nuclei yields show a similar trajectory, with nuclei ranging from 7,000 nuclei per milligram of tissue to hundreds of thousands of nuclei per milligram, again, depending on the expected cell density of the tissue. And now with our specialized NIC Plus cartridge, you can obtain thousands of nuclei from tissue samples as low as 2 milligrams in size. And we just discussed that debris could cause a run to fail or show up as background in the single cell data. So at S2, we have a simple solution for this. Our nuclei debris removal reagent allows users to remove debris from their sample through a simple centrifugation step. This process is quite fast. It's around 10 minutes total. Here, you can see a sample was run through a cell cytometer before and after our debris removal step. On the left, you see that around 39% of the events were DAPI stained nuclei before debris removal. On the right, you see that around 90% of the events were DAPI stained nuclei after debris removal. This reagent has been a fan favorite, especially when dealing with myelin debris from brain isolations. The Singulator platform is highly versatile, compatible with over 80 sample types across various organisms, including human, mouse, rat, chicken, and zebrafish, and many, many more. This list is ever growing and can be found on our website. So we offer 3 different Singulator instruments, and each is suitable for the needs of our individual users. Each instrument is capable of automating isolation of nuclei or cells from fresh, frozen, and OCT embedded tissues. For full automation of FFPE nuclei or cells, including the deparaffinization and rehydration process, that is capable on the Singulator 200 Plus. With the Singulator 100, you can process 1 sample at a time for a total of 8 nuclei isolations per hour to give some kind of, time point. You can double that with the Singulator 200 and 200 Plus by processing 2 samples simultaneously or independently for a total of 60 nuclei isolations per hour. You can also process 2 FFPE samples on the Singulator 200 Plus in 40 minutes. So the entire deparaffinization to rehydration, your washes, and then your nuclei or cell isolation from your FFPE samples can be conducted in 40 minutes. The Singulator 200 is now available for pre-orders and will be launched in 2025. So if you have any information, feel free to reach out to Techtum and they can get that pre-order going for you. I want to kind of talk about the reproducibility and precision across various sample types using the Singulator, so that way you can see in real time the Singulator performance. So the Singulator is designed to deliver reproducible and precise results across not only clinical samples, biological samples, or technical samples, but many different samples, ensuring reliable, high-quality data. Here, we show reproducibility and precision in clinical samples processed on the Singulator. Cells prepared from 3 human normal lung biopsies from 3 different lung cancer patients demonstrated isolation of the expected cell types across the 3 samples. Similarly, we show reproducibility and precision in biological replicates processed on the Singulator platform. Nuclei were prepared from 3 mouse cerebellums from 3 different mice, demonstrating isolation of, again, the expected cell types and uniformity across the samples. But in the interest of saving time, I didn't divulge into those 2 different sample sets, but for this meeting, let's discuss technical replicates a bit more in detail. A mouse kidney was divided into 6 parts and processed on the Singulator using the NIC+ cartridges. Results showed high reproducibility and demonstrated isolation, again, of the expected cell types and uniformity across the 6 technical replicates. We were even able to see a rare and fragile podocyte population. And how we did this is that we dissected out a single kidney and divided it into 6 pieces. Then we ran those kidney fractions on 2 different Singulator 100s and 2 different Singulator 200s using both bays for a total of 6 samples. We followed the standard nuclei isolation protocol. We used the NIC+ cartridges and the nuclei isolation reagent, storage reagent, and RNase inhibitor. Since kidney has low debris, we did not use any debris removal or any sort of cleanup or enrichment downstream of the isolations. These nuclear suspensions were counted and they were put into 10x as assay. Libraries were sequenced on the Illumina sequencer and the data was processed with Cell Ranger using Seurat. What we saw was that each sample had high fractions of reads in cells, which is a result of low ambient mRNA contamination percentage across all 6 kidney fractions. Upon downsampling of the libraries to equivalent reads per cells, we see reproducible performance across the 6 kidney samples. The 6 kidney samples exhibit comparable gene expression profiles per cell, consistent UMIs, and demonstrate low levels of mitochondrial contamination. Finally, we wanted to see the correlation amongst these 6 samples. So we did a Pearson correlation where we conducted pseudo bulk average gene expression across the individual samples. So we had 15 different pairs of comparisons amongst the 6 samples. Here are just 2 representative Pearson correlations. On the right is the worst correlation of all 15 pairs. So even the least well-correlated sample set had a Pearson correlation of 0.985. Every comparison was above 0.985. So this indicates that the expression patterns of genes in these 6 samples are highly similar, with little variation between them, confirming that the Singulator platforms are in fact reproducible. The Singulator platform has significant impact on research workflows. Many researchers reported improved precision, efficiency, and reproducibility. And the biggest takeaway is that the Singulator is so easy for them that they will never go back to manual methods. The Singulator is increasingly cited in scientific publications, supporting research that spans again, disease mechanisms, therapeutic target identification, and multi-omic assays. So it's clear that the platform is making a real-time difference in advancing scientific discovery. The platform is proven compatible with various protocols, sample types, and research goals. It's automated, easy to use setup ensures high quality results and minimizes that risk of human error, making it ideal for many single cell applications, such as BioSkryb. So with that, I would like to hand it off to John, who is going to be our next speaker from BioSkryb.
All right. I'm going to share my screen. Thank you for inviting me to speak here today. So my name's John Bond, and I'm the head of support at BioSkryb Genomics, and I've been here for about 3 years now. And today we're going to talk about a revolution in single-cell genomics and transcriptomics featuring ResolveDNA and ResolveOME. So I want you to understand with this presentation who BioSkryb is and what we do, what we offer, types of products we offer, the general workflow, and we're going to get into the technology a little bit, and also some case studies. So show you the applications and some real-life case studies. And then I'll give a summary and be happy to answer any questions. And this talk is really meant to stimulate discussion, so be happy to answer any questions at the end. And also, if you want a future meeting with me, and I'd be happy to get on a call with Techtum and you to discuss the technology and how we could fit with an application in your lab. So as Nabha said in her great presentation in the beginning, we'd like to set the stage and just talk about the great heterogeneity in just one being, like just one human. There's over 30 trillion cells, and most of those cells have a genome with 3 billion base pairs of DNA transcribed into tens of thousands of mRNA molecules and tens of thousands of proteins. Just for a single cell, it's complex, and there's cell-to-cell heterogeneity, and that cell-to-cell heterogeneity is really important to understand, especially in diseases like cancer and neurological diseases, etc. And we think at BioSkryb, we provide products that could resolve that heterogeneity at the genomic and transcriptomic level better than other technologies. And we could also look at targeted cell surface proteins as well. So the two products that we have to look at the cell-to-cell heterogeneity are called ResolveDNA, which is whole genome amplification. And you could get the whole genome amplification with library prep. And ResolveOME. And ResolveOME is whole genome amplification married to full-length transcriptome, and also includes library preparation for next-generation sequencing. The workflows are very simple. They take a half a day and maybe on one day, half a day to 6 hours on one day, and maybe 4 hours on the second day, and then you're ready to sequence. So very easy workflows. There's manual and automated workflows to make life easy in the wet lab. We also have, as Techtum does as well, we also have a Resolve Services department. So we could go end to end from cell isolation and enrichment all the way through analysis. And we do use S2 Genomics, for example, to isolate nuclei, to use our technology in front of our technology as well. And we'll talk about that in a little bit. But we could do anything just like Techtum can from end to end using our technology all the way through analysis. And we have a software tool called BaseJumper that could help with your single cell analysis as well. Now, with that little bit of background about who BioSkryb is and what we do, I'm going to get into how we're different from other technologies and talk about the core technology that powers both ResolveDNA and ResolveOME, and that's primary template directed amplification, or PTA. And this is a method of whole genome amplification, and it uses 529 polymerase. That's represented by the purple blob that you see on the template DNA, which is blue. And we use random primers, which will interrogate the entire human genome, for example. They're random primers. It's not like PCR where you use opposing primers. So we use random primers that'll interrogate the whole human genome. I do want you to realize that it'll also amplify bacterial genomes or mitochondrial genomes. So in that human cell that has mitochondria, you'll also get the mitochondrial DNA amplified. And with ResolveOME, you'll get the mitochondrial RNA as well. So just be aware of that. And we use an isothermal polymerase. So the reaction occurs at 30 degrees. So again, no need like traditional PCR to heat the sample up to 95 for each cycle. There's only one cycle. It's just 30 degrees for a certain period of time. We also add these irreversible terminators that will stop the reaction short and create a distribution of fragments anywhere from a few hundred base pairs to 5,000 base pairs, with an average in a human or mouse cell of about 1,200 base pairs. And this irreversible terminator is what allows for whole genome coverage from single cells. And this is very powerful. Also, the polymerase that we use has a higher affinity for longer pieces of DNA than shorter pieces. So it does not go back and amplify frequently the daughter amplicons. It'll go back to the primary template and amplify it. And this is very important on the analysis end. If there's an error caused by the polymerase, it won't get propagated over and over again. Also, this is not exponential amplification like it is with PCR. It's a quasi-linear form of whole genome amplification. And what I show here on the right side of the screen is an example of human chromosome 1 from PTA, primary template directed amplification, and MDA, our closest competitor that you may or may not be familiar with, which is another whole genome amplification method. And with PTA, we get good even coverage across the whole genome and both alleles amplified. We typically get 95% or greater of whole genomes covered from single cells. With NDA, you get 40 to 70% of the genome covered. Then with the next cell, a different 40 to 70% of the genome covered. So you get a lot of allele dropout. So with PTA, because the coverage is so great, not only can you look at copy number variation, but you could also look at single nucleotide variants and structural variants from the same experiment, from that same single cell, just based on how deeply you sequence. So with that little bit of background, let's go into the workflow for ResolveDNA, and then we'll go into the workflow for ResolveOME in a little bit. So the formats are, this is a plate-based format. So with BioSkryb, it's not digital droplet, it's plate-based. So you would get your cells in wells. There's 96 cell formats or 384 well formats. So you need to get your single cells in wells, and using FACS machine is the standard way to do that. So you can enrich with cell surface markers and use FACS to get the cells in wells. So the cell collection piece, and I'll talk about in a few slides too, and bring up S2 again, is very important because our technology works best with fresh frozen or fresh cells and not fixed cells. After the cell collection is done and the cells are in wells, then our technology is employed. So a cell lysis is done first, and this will lyse the nuclei and access the genomic DNA. The whole genome amplification is done by PTA. And you could, if you purchase our library prep, that would be next. But the great thing about the BioSkryb PTA technology, our library or our whole genome amplification method is agnostic to the library prep. So if you want to use Illumina, you could use Illumina library prep. You could use our library prep, which is the ligation-based method, Illumina-based method. Customers have used Oxford Nanopore, customers have used Singular, customers have used Ultima, PacBio, and we'll talk about some of these later, but you could use other methods to sequence, not just Illumina. So just realize that as well. So we also have ResolveOME. ResolveOME is very powerful because not only do you get the power of primary template directed amplification, you get the whole genome amplified, but you also get the full length transcriptome. This is not 3' or 5' end counting. This is full length transcriptome. So you could look at gene isoforms. You could look at a cell and see that P53 is not expressed in that cell, and then go back to the DNA from that same single cell and see that there's a copy number loss. And we actually, I'm going to show, talk about a study, a breast cancer study later that discusses that, for example. So very powerful, look at gene expression, but also look at the whole genome from the same single cells. So what are some of the applications that you could do with our technology? So there's hem-onc applications, and I'm also going to bring up, because the ASH meeting in San Diego is coming up next week on December 7th. So some of you may be going to that meeting. There is an abstract looking at multiple myeloma patients that are relapsed and refractory, and isolating circulating tumor cells from their blood, and then using our Resolve DNA technology to amplify whole genomes and sequence that DNA to look at to have more informed decision on those patients' future treatments. So that abstract is available if you go to the ASH website, and you could have a look at that abstract. It'll be presented at the ASH meeting, and it's not from BioSkryb, it's from our customers. So there's solid tumor applications, and I'm going to talk about the breast cancer study that we're doing with the Duke University Medical Center from Shelley Wong and Jeffrey Marks. And this is an example of 4 cells looking at copy number, and you could see some copy number losses in 3 of the 4 cells, and they happen to be in areas where there's tumor suppressor genes like RB1, TP53, and BRCA2. Neurology is a very big application, and just even looking at normal cells, so looking at neurons in the same patient that are normal, and looking at the cell-to-cell heterogeneity or tissue heterogeneity from different parts of the brain, has been done and it's been published, as well as comparing diseased neurons to normal neurons, as well as looking at age. So from infants, adolescents, adults, and age individuals, whether looking at normal neurons over time and looking at looking at mutation burden in those normal neurons, or comparing, for example, Alzheimer's to normal cells and looking at the mutation burden in those cells. These have all been published papers that you could find at a resources page on our website, and you could download the links to download those publications. An up-and-coming space for us, because we get the whole genome amplified, and with ResolveOME, you get the transcriptome. If a genetic element is put into a cell using cell and gene therapy methods, using CRISPR or viral vectors, for example, you could look and make sure that the genetic element is there, so on target, or if it's off target, you could do both on and off targeting, because we're getting the whole genome amplified. And if you have a genetic element that needs to express a functional messenger RNA molecule, you could look at that as well with ResolveOME. So you'll get the whole genome amplified, plus you could detect that molecule, that expression with ResolveOME. And we have a 10-year deal with Cooper Genomics, for those of you that didn't know, to look at pre-implantation genetic testing. But this is also a growing area where to look at embryos before they're implanted for pregnancy. You can look at a few cells and look for aneuploidy, PGTA, for that embryo, and also look at PGTM, which is monogenetic disorders. Before that embryo would be used, you would know that information. Now, to get more into the workflow of what we do and also into ResolveOME before I talk about the breast cancer study, it's really important, as Nabiha said, to get good material to start with. So that garbage in, garbage out that you always hear in biology and biotechnology, that really applies with our technology as well. So you want good cells because we're trying to get the whole genome amplified as well as a full-length transcriptome. So you want good cells to start with. So S2 Genomics is a really great platform to get good cells and good nuclei. And we use for nuclei prep, we use S2. We have the instrument in-house and we use it consistently to isolate nuclei before we do ResolveOME, Resolve DNA. So sample preparation is very important. The FACS is very important. As Nabiha said too, viability is very important. So another system that works well is the Levitas, or you could just do dual viability staining with propydium iodine and calcium AM. That's what we use mostly internally, but we also use other means to check for viability. The viable cells will produce the best data. So very important upfront to get, you want good healthy cells or fresh frozen cells for our technology to work the best. And then you can employ the Resolve DNA and ResolveOME technology, the whole genome amplification, and with ResolveOME, whole genome amplification married with full-length transcriptome, sequencing, and then analysis. Using base jumper, you could use your own common methods for analysis as well. Another thing we could do is on the ResolveOME side is we could rinse the cells. We could wash the cells with the BioLegend Total-seq A. Antibodies or custom antibodies, and that data for the cell surface proteins would come off in the RNA arm of ResolveOME. And I want to remind everybody that other sequencing methods have been done as well with our technology, including Singulator, not just Illumina, but Singulator, Ultima. There's a bioarchive with Oxford Nanopore, PacBio, which we'll talk about a little bit later. Element has been used, etc. So just realize that. Now the ResolveOME workflow, and then we're going to go into the breast cancer study. So again, the cell collection piece we already spoke about. It's very important. And again, we have a 96 and soon to be released 384 well reaction flavor of ResolveOME. So what's done after the cell collection? The cells are in the wells. Is a cytosolic lysis is done first to access the poly-A messenger RNA. And we use an oligo DT primer with a biotin group attached to it to make first-strand cDNA. And that biotin group is going to become important later on. Then we do the primary template-directed amplification. So the nuclear lysis is done, and the whole genome is amplified by PTA. Now we have DNA and cDNA in the same tube, and the cDNA has biotin on it. So we add a stragavidin magnetic bead and a magnet, and all the cDNA is going to pellet to the magnet. The supernatant will contain the DNA. It gets put into a separate plate, and then a next generation sequencing library is made. And all the reagents to make the library and do this procedure is contained in the kit. The cDNA gets amplified off the beads, and then a next generation sequencing library is made with the cDNA. And they have different barcodes, so you could sequence them together on the same flow cell, and then you have your data analysis next. So that's how ResolveOME works. And now we're going to get into the primary breast cancer study, which uses both Resolve DNA and ResolveOME to look at copy number variation, single nucleotide variants from the same cells. So in this study, we're up to now, this is an ongoing study. It's been going on for quite a while now, and it started out with one patient, and now we're up to actually more than 12 patients, but I'm going to show a little bit of data from some of these patients. And you can see every row represents a patient here, and they have different phenotypes. Some have ductal carcinoma in situ, some have invasive ductal carcinoma. The whole goal here was to learn more at the genomic and transcriptomic and also cell surface protein level of what's going on from a patient that started with DCIS but then got invasive breast cancer. So they want to understand it, and also the tumor microenvironment. And they looked at about 80 cells per patient for 960 cell total with this group of patients. And Shelley Huang and Jeffrey Marks, these are the people working on this. And some of this work has been published in bioRxiv. Eventually, it'll be a publication in a journal. What I'm showing you here is some of the data from the copy number variation. So this is from left to right, you have the whole genome from chromosome 1 all the way to the sex chromosomes. And we have 757 rows. Every row represents a single cell. Every time you see red would be a copy number gain. And when you see blue, there's copy number losses. You can even see subchromosomal aberrations with our technology. Again, you're getting the whole genome amplified. So this is across the entire genome. And in the bottom set of cells, we have the control cells, but there's not cancer cells where there's less copy number variation going on. And at the top, we have a group of cells that are cancer cells. And one thing I want to mention as well, as I talked about before, some of the cells had losses in P53, in RB1, in BRCA2, so tumor suppressor genes, which would make sense. And also looking at single nucleotide variants, many of the cells had a PIK3CA variant that's common in breast cancer. One patient I want to mention, patient 4, and that they were diagnosed as HER2 negative and were put on a treatment. And when we looked at the cells, the single cells, we looked at about close to 60 cells. We saw 2 of the cells had a deleterious mutation in HER2, which would have, if the oncologist would have known about this, would have changed the course of treatment. And that's what this is all about at the single cell level. Now, of course, 2 out of 60 cells, this is not clinically relevant. There is no basis for this right now, but in the future, we're hoping that there will be, that single cell will be employed, because this data would have gotten washed out with bulk sequencing. You would not have seen it. So very important to realize and recognize that the power of single cell technology. We also did a principal component analysis, many of the cells that were normal or different cell types, or that had the PIK3CA variants and the tumor suppressor genes were lost. And you see they tend to cluster together. So the diversity of cell types you could see in the bottom right. They have different colors, the epithelial, the endothelial, fibroblast. They have the different colors and they all cluster together. They're more of the normal cells. The cells that had the PIK3CA deleterious variants that are common in breast cancer clustered together. And also the normal cells clustered together. But we did recognize too that there's this one cell, because we have the RNA as well as the DNA in the same cell, that seems to be going through a transition into a more invasive type of cell that could potentially cause to go from from DCIS to a more invasive breast cancer. This is the type of work that can be done when from the same single cell you get the DNA amplified as well as the full-length transcriptome. I have a couple more slides. One, I did mention earlier that there's hem-onc applications as well. So there's going to be an abstract at ASH from a study that's done with multiple myeloma patients. I think there's about 16 patients, almost 500 cells were looked at. And what this group wanted to do is these patients were relapsed and refractory, and they wanted to do a blood draw and isolate circulating tumor cells and sequence, whole genome amplify the DNA from those cells, sequence those cells, and then hopefully have a more informed decision on how to treat and why they're refractory, and then how to treat these patients. So that's what this abstract is all about. And they used our ResolveOME technology to do the whole genome amplification after they did these blood draws. Also, they want to be able to do this because to replace, hopefully in the future, a bone marrow biopsy, which is much more invasive than just a blood draw. So if you could isolate those circulating tumor cells, and there's not many of them, and then sequence the whole genomes of those cells, you can learn a lot more about the disease, that MRD from that disease, and potentially change the treatment for the patient in a good way and extend their life. So that's going to be at ASH next week. On December 8th, it's going to be presented if you're going to the ASH meeting in San Diego, California. And I do want to say that the future, we do have there's a bioRxiv paper that just came out a couple weeks ago, which is with a different PTA kit. It's not available for purchase yet, but in our services we could do it. But long read PTA, so it's a modified PTA reaction, and you could do long reads with the with PacBio system. You can also use PacBio. People have done it with our standard PTA kits, but this is the ability to get even longer reads than that 1,200 base pair average that I talked about. And it opens up the door for other applications as well, be able to do that with our technology. So this has been done and it's been published, and we know how to do it. And it will be available in the future, and it will be available shortly in our services group. With that being said, I want to give a nice summary here. So we have two products. We have the ResolveDNA and ResolveOME, both powered by PTA. We also talked about that BioSkryb also offers services just like Techtum does. And the flavors of our kits are 96 reaction and 384. So they're plate-based reactions. So you have to get cells and wells using FACS or some other means. And our technology works best with fresh or fresh frozen viable cells, because we're trying to get the whole genome amplified and the full-length transcriptome. We can detect, because our technology is so sensitive and we get whole genome amplified, we can detect single nucleotide variants from single cells, as well as structural variants and copy number variants. And we could also look at gene expression from that same single cell, look at isoform detection because it's full-length transcriptome, not just end counting, and also cell identity to identify the cell, if it's an immune cell or a cancer cell, because we're getting the RNA as well as the DNA. There's many different applications, including oncology-based, so mutation discovery, or MRD that I just talked about from that ASH abstract. If someone already has cancer and there's a longitudinal study being done, you can look to see if the cancer comes back, or you can learn more about if someone relapsed, more about their cancer by isolating those circulating tumor cells and looking at the DNA and the full-length transcriptome. Neurological applications we discussed to look at disease cells versus normal cells, or just look at normal cells over time and look at the mutation burden that's accumulated in those neurons over time. Pre-implantation genetic testing. Again, we have a deal with Cooper Genomics. They use our technology, the ResolveDNA, to look at PGT-A, which is aneuploidy in embryonic cells, and also PGT-M, or monogenic disorders they're looking for on the PGT side. Of course, there's other applications along in these lines as well. I didn't talk a lot about microbiome, but I did bring it up that you could also amplify bacterial cells. We use that random primer, so we did discuss that. So you can amplify the DNA from bacterial cells and sequence it. Also, low input biomass. You don't have to have a cell at all. You could just start with nucleic acid. A wastewater study was just done in Europe over time, over a 17-month period, where they looked at wastewater and they found known viruses that were in the wastewater that are pathogenic, as well as unknown. So you could do those types of things with our technology as well, and just start with nucleic acid you might not even know the concentration of, and then amplify, make libraries, and sequence. And we do have a base jumper software as well to help with analysis. We offer that as a service, or you could download Base Jumper local if you have any appropriate system and do the analysis yourself. With that, I'd like to thank Nabaha and Hakeem and all the other people at Techtum. Thank you so much for letting me speak today, and we'd be happy to stay on and take any questions. Thanks so much.
Thank you, John, and thank you, Nabia, for for taking the time today and for presenting the solutions from S2 Genomics and BioSkryb Genomics. And thank you everyone for joining the webinar today, taking time for it. We now have a few minutes for questions. If there's any questions from anyone in the chat, you can either write your question in the chat or raise your hand and then unmute yourself if you wanna discuss with the presenters today. We will also, of course, we'll share the recording of the webinar, and any questions we are also happy, of course, to schedule another meeting or discuss via email as well. So one question is, would BioSkryb be compatible with exome enrichment?
Great question. So will BioSkryb, is it compatible with exome enrichment? Yes, it is. And there's actually a couple publications using IDT XGen, and there's one using an Agilent SureSelect that's available now. Those bioRxiv publications. We're working on our own exome enrichment system, but we are, again, agnostic to it. So Element has a system that we know works with our technology. And I'm forgetting another common one, IDT. I already said IDT XGen, I said Agilent. Any of the capture methods will work well. So you make the library first, and then you do the capture afterward. And we do have some data from customers that's already been published. Great question. So that'll allow you to do much less sequencing. So instead of deep sequencing you might have to do if you're doing discovery to look for single nucleotide variants, you could do 10-fold to 20-fold less sequencing if you're only doing the exome, right? So just 20 million reads per cell, 2 by 150.
Thank you. And I have a question as well for you, Nabiha. So can you freeze down nuclei samples post-isolation?
So no, there is no recommended method in order to freeze down nuclei from a nuclei isolation. There are methods of fixing it. So, people use methylation or not methylation, methanol to fix their nuclei. They also use other kits available on the market to fix their nuclei and then process at a later time. But you can't just simply take any nuclei suspension, whether it's isolated on the Singulator or in a manual method, and just simply freeze it like you do with cells.
Okay.
Just to add on to that, when we're using the S2 Singulator, we usually go right into fans. So again, we're using the fax machine, and right after we do the S2 Singulator to isolate nuclei, for example, we go into the fans protocol and deposit the nuclei into wells. And it works great. Get really good data.
Great, thank you. And we have a raised hand. Asade, you want to ask a question?
Yeah, hello, and thank you for a great presentation. Impressive. I've got a question from Nabiha, S2 Genomics. There are basically two questions. You probably mentioned it, I missed it. So for Singulator 200 plus, is deparaffinization and rehydration the whole procedure? How long would that take from the time that you put the sample in and out?
Yeah, so from the time you place the sample in and to the time that it comes out, it takes around 40 to 45 minutes. So you are able to process 2 samples simultaneously on the Singulator 200 plus at a time and get it within the hour. So I'm talking about your counts, your downstream cleanups, and then loading it into whatever downstream assay.
Impressive. So that means that as soon as you take the sample out of the cartridge, the green, I assume, and you put it on the NIC Plus cartridge, then like 10 minutes extra usually, and then so in less than 1 hour, A to Z would take.
Yeah, in less than 1 hour, you will get 2 FFPE samples processed on the Singulator 200.
Wow.
So that entire the deparaffinization, rehydration, the various ethanol washes that it conducts, all of that, and then your nuclei isolation, So within the hour, you get 2 samples coming off of the Singulator.
Wow, that saves a lot of time and money and human power.
does.
Very impressive. Another question is about the cell isolation and the cleanup. Is there any solution for clean up the cells? Because I assume the one that you said was for the nuclei, right? Or
Right. So we have the nuclei debris removal reagent. We do not sell a cell version of it. We do have recommendations though
I
on
see.
what to do. So if anyone were ever to be like, hey, I'm doing a cell isolation, it's very dirty, especially when you see that in tumor samples. We have recommendations on what you can do. Obviously, nothing beats FACS sorting, right? But there
Yeah.
are other options that we recommend that work really well with cells.
Excellent. So my concern about particular sample is like red blood cells and so on.
Yeah, red blood cells, we do what's called an ACK lysis, which is just a standard red blood cell removal
see.
method in the
Okay.
market. You can either make your own ACK lysis in-house, or you could purchase them through multiple vendors. It's quite cheap, and it works really, effectively for all red blood cell lysis methods. So that's what we recommend, and that's what we do as well.
I see. Many, many thanks.
Yeah.
Great. Thank you so much. And John, one more question for you. So how many cells would you consider to analyze as a first pilot experiment?
We'd have to discuss your project to see, but most customers will do, at least a plate per sample. So like a 96 cell, per sample, or you could do 48, if it's just a, pilot, you know what to expect. And we could take that pilot, whether, we could discuss if you're going to do it with Techtum or do it with us, but we could take that pilot, anywhere from you send us the cells and we'll do the FACS sorting all the way through to low pass sequencing. And when you do the low pass sequencing, it'll give you enough data. You'll get your copy number analysis data as well as your full length transcriptome, which is a couple million reads per cell. So it'll give you enough data. You can look at that data and decide which cells to choose on the DNA side where you want to sequence deeper and look for single nucleotide variants and structural variants, for example. So the copy number data you get with just a couple million reads per cell. If you want to look for single nucleotide variants or structural variants, you have to sequence deeper. And you can look at the data from the low pass sequencing data to determine which libraries you're going to do deep sequencing on.
Okay, great. Thank you so much. And I think there's no more questions. I think we've been around 1 hour, so I think we'll end it there. Sorry for the technical problems in the first few minutes, everyone. And thank you so much again, John and Nabiha. And thank you for everyone who joined. And we can send out any material or set up any meetings post this webinar to anyone who's interested. So if you're in the Nordics, please reach out to us at Techtum, and otherwise reach out directly to BioSkryb or S2.
Before we end, I would like to, if you don't mind, mention that we are having something called the Singulator Sessions here in the US. They're a webinar series. We already had one, so I gave the first one last week. And we have our next one next Tuesday. So if anyone wants to join those sessions, we're going to talk about oncology applications and neurology applications in the next 2 sessions. So if anyone wants to join, it's open for everyone. All you need is a Zoom account in order to join. So feel free to join that and learn more about the Singulator platform.
Thank you. Yeah, it's a good idea to follow both S2 and BioSkryb on LinkedIn as well to see any news, I think.
Yep.
Okay, then
Thanks so much.
End with that. Thank you so much, everyone. Take care.